Hello, everyone. I'm Sahit Totamgari. I'm a cardiology fellow at LSU Health in Shreveport. Today, I have the pleasure of introducing Dr. Vemulapalli, who's Associate Professor of Medicine at Duke Health and a member of the Duke Clinical Research Institute. We are going to discuss today about Dr. Vemulapalli's research on adaptive AI for cardiovascular event adjudication. Welcome, Dr. Vemulapalli. Thank you so much for joining us here. And it's an honor to have you here.
Thanks for having me. Excited to talk about it.
Awesome. Thank you. So I'll start off by saying, the intersection of AI and clinical trial methodology is one of the exciting frontiers now. Can you, give us a brief summary of what your research is about and what question you set out to answer?
Yeah, sure. So as you said, I think there's a lot of excitement in this field. There's a lot of areas of clinical trial research that need to be modernized, both for efficiency purposes, but potentially also for reproducibility and accuracy purposes. So we chose to look at one of those things here. One of the important parts of a clinical trial is ascertaining events. And the gold standard for doing that is something called event adjudication, where you basically have a group of experts sit down based on a predefined definition of an event, review the medical records of that particular participant, and then decide, did this event happen or not? The FDA and other regulators and clinicians rely on the fidelity of this process to interpret a clinical trial. So what we did was we said, can we train an AI algorithm that can actually do some of that work to identify events? And so we actually took events and adjudication data from two trials, one called Odyssey Outcomes, which was a randomized trial of alirocumab. And we trained a model to identify two events, specifically MI and stroke from that particular data set. And then we said, listen, can we validate that in an independent data set? And that was a different clinical trial called Euclid. And that was a trial of ticagrelor versus clopidogrel in patients with peripheral artery disease. And the interesting thing that we did there is we said, okay, can you identify MI and stroke in Euclid? But the definition of MI and stroke in Euclid was slightly different than in Odyssey. And then we also said, can you also identify other events like cardiovascular death and bleeding in Euclid, which it had not been trained on in Odyssey? And furthermore, we said, okay, how does this compare to the original human adjudications? How does this compare to if we did a hybrid approach where we use our AI model for most of the adjudication, but for the ones where it's uncertain, then we say we'll have a human do it. And then the third method we used was we just compared it to GPT-4. So GPT-4 is ChatGPT. So how does our model do against ChatGPT? And what we found was that this was pretty successful. Now, the version of this that did the best was the version where the model adjudicates, but then where it's uncertain, we have a human being do the adjudication. So we used the 30% that it's most uncertain on and gave that to human beings, but everything else it adjudicated itself. And we compared that to GPT and it was better than GPT-4. And even the model itself, where it does all of them, was better than GPT-4. And then we said, okay, the proof is in the pudding. So what if we try to re-estimate the trial results, the primary result of Euclid? So the primary result was cardiovascular death, MI, or stroke. And we were able to re-estimate it. Now, in that particular trial, the overall finding was no difference between ticagrelor and clopidogrel, and that's what we found. And the hazard ratio was very, very close to the original hazard ratio. And additionally, when we look at the effect of clopidogrel versus ticagrelor, not just the overall effect, those were also very, very similar to the original trial.
Awesome. That's thank you so much for answering that question. It's a great foundation. So that leads me to my next question. So you spoke about Odyssey outcomes and the Euclid trials. They're quite a different trials and different populations, different events. So what made these two trials the right sandbox for you to test the adaptive AI model?
Yeah, so a few things. One is they had different definitions. So we wanted to make sure we had different definitions. The second thing is they're both fairly large trials, north of 10,000 patients. And so that gives us a lot of events to both train on and potentially validate on. And then additionally, we're cardiologists, so we wanted cardiovascular events, right? So we started with MI, stroke, cardiovascular death, bleeding, and of course we have plans to extend it to heart failure, acute limb ischemia, other things that would be of interest to the cardiologist.
Awesome, thank you. So, talking about the outcomes that you looked at, MI and stroke, right? When we talk about the MI classification, the distinction between the type 1 and type 2 MI is, very subjective, even among expert adjudicators. So, even in Odyssey trial, I think about 17% of the events were type 2 MI. So, how did the model handle that subjectivity?
It's a great question. So first off, I would say when we went into this, we were thinking, okay, let's see if we can get MI right. Yes, no. We weren't sure if we could even do that. Looks like we can. And then we said, okay, can we get type 1 versus type 2, knowing how difficult this is? And in fact, type 1 versus type 2, the differentiation was very good. Now, the question is, what is very good? So this is a key fact. If you take a set of events and get one CEC, that's Clinical Event Adjudication Committee, to look at those events, and then you get another CEC to look at it, or you get the same CEC to look at it again in a blinded way, they're going to be off by about 10 to 20% of the time. So that's the fidelity you're aiming for. We can't do better than what the humans are doing because we're training on the humans. And so we found that differentiating type 1 and type 2 MI was similar to what human beings were able to do.
Okay, that's awesome. That's really great. So talking about the MI again, if I've looked at the title, something that drew me was the changing definitions on the title. So let's say when you have something like evolution from conventional troponin to a high sensitivity troponin assay, shifting the threshold, how does an adaptive model distinguish between an increase in the clinical event versus what is essentially just a reclassification artifact?
Yeah, so that's a great question. So let me take one step back before I answer that, which is that when we expose the model, what we do is we essentially tell the model what the definition of the event is, and that's different for every trial. And then we give the model the dossier, which is the collection of all the documents of that participant from which it has to determine whether an event occurred or not. So when we then expose it to a new trial, we give it just a few cases, in fact, 10 cases and 10 not cases to learn on with the new definition. So it cannot differentiate why there's a difference. Like if you change the threshold of a troponin value, that's obviously going to change how many MIs you have, and it's going to be beholden to that. So if your definition says a troponin has to be over the 99% of upper limit of normal, it's going to adjudicate that way. It doesn't know what the upper limit of normal is, except for what's in the documents. So it cannot tell you, oh, this difference is simply because you changed the threshold of the measurement.
Awesome. Okay, that's really fascinating. Thank you for answering that question. So, one of the other biggest questions audience might have is, the generalizability of this approach. Can we use it for other events, other patient populations? So, where are we in terms of that?
So that was, in fact, exactly why we looked to have an adaptive model, because we said, listen, for the purposes of clinical trials, it's not very helpful if you have to train a new model every single time you have a new clinical trial, right? Because it takes all the data to train it. So you need to be able to show that you trained a model and then you can use it again and again and again. And wouldn't it be even better if you could use it with just a little more training on a different event type? So I think in our study, by going to different event types with a little bit of training, we have shown an ability to generalize. Now, we've shown it just amongst MI, stroke, CV death, and bleeding, but we believe, and we will be doing further work at looking at other endpoints. Now, I will say that's just in clinical trials. I actually think that the utility of something like this goes well beyond a phase 3 trial. It goes into phase 4 or real world evidence. So what is it that prevents us from using CEC when we do real world evidence? Well, it's timely, it's costly. So, it's, because you have to have a human being reviewing everything. So what if you could actually use these algorithms to identify events in the EHR? Now you could run a trial through the EHR, or you can run an observational cohort through the EHR, And I think this is possible because essentially the data that goes into the dossier, the vast majority of it is EHR medical records.
Right.
So that's where we're looking in the future for this.
Awesome. That's very promising future there. So, what would it actually take, for the FDA to accept, an adaptive AI model as a primary adjudication? Are we still in that? Are we there yet, or are we just in the era of AI supplementing, like you said, to human CCs?
I think we're part of the way there. So what I think we actually need, everything I've described to you is retrospective. We train a model on existing data, we apply it to another data set that exists, and we say, oh, we can reproduce what happened. But I think what we really need to do is say, let's take a trial that's about to start or has just started. Let's adjudicate it two different ways. Right? Let's adjudicate it our traditional way with a group of humans. Let's adjudicate it now using the AI model, probably in a hybrid approach like I described, where the uncertain ones go to human beings. And then let's compare. And specifically, the goal is not to say that the AI model with humans for the uncertain ones is exactly the same as a separate CC group that's only humans, because we know that if you take two human groups, they differ by 10 to 20%. So we need to get to that level, 10 to 20%. That is what I think we need to show for regulators, and more importantly than regulators, for clinicians who read these trials afterwards to feel confident and comfortable.
Awesome. That's really fascinating. So one last question, and I ask this as a fellow myself. So what career advice do you have for fellows in training and early career physicians, not only for who are interested in the realm of AI, but, in general?
So I would say, and this is going to sound a little cliched, but you need to do the things that make you excited. And so this was exciting for me. I found a collaborator at Duke to help work on this with me. And I get up in the morning wanting to do this. So when I was working on the paper for this at midnight, at 2:00 in the morning, it's because I'm excited about it. If I wasn't excited about it, I wouldn't be doing it. So find the thing you're passionate about and excited about, and you'll be happy with it.
Awesome. Thank you so much, Dr. Vemulapalli. That was a pleasure talking to you. Dr. Vemulapalli is presenting his research as a part of Investigative Horizons tomorrow. So please do check it out. And please do check out the X and YouTube channels on Fits on the Go for more content like this.
Thank you.